recap of what happened. I think we stopped here. The inhibiting substance that is used in MSA is basically 7.5 NaCl or sodium chloride, and then the test used to differentiate mannitol fermenting staph species would be novobiocene test, and non non-mannitol fermenting rather. So we're testing for epidermidis and saprophyticus. All right. So please don't forget our mnemonics, SIR and SSR, which basically means staph epidermidis is susceptible. SSR, staph saprophyticus, is resistant. All right. Clear? Everybody clear? All clear? Cesar, is it clear to you? Okay. So I hope it's clear for Cesar. All right. Because yeah, and Noreen and yeah, and Angel. Hopefully it's clear for you guys. All right. Now let's move on to catalase test. So catalase test, I think, is another way to differentiate what is another differential test or another presumptive test for differentiating staph and strep species. Okay, so staph is basically going to test positive for catalase test, and you're going to use what reagent again? 3% hydrogen peroxide. Okay, and the positive result is of course bubble formation. And if we want to sound posh, we can use the term effervescence, o 'di ba? It sounds like a band from the 90s, like Evanescence. I think it's a band from the 90s. I'm not sure. I'm not also familiar with bands, but yeah, um, let's go with that. But anyway, I don't know if people are still. I don't know if there's a generation gap. Kung ganun natin go, yeah, I think that's how far our generation, each other's generation is from each other. But I always remember the band Evanescence every time I open, every time I see the word effervescence. So hopefully you guys will also have some sort of way to remember this. All right. Now, I want you guys to remember what I want you guys to take from this particular slide is the species of organism that will test positive because it's a pretty common test, especially in most hospitals who don't rely on automated bacterial identification. So during workups. When I say workups, um, these are basically your tests in which you, as medical technologists, will have to think critically and investigate what are the characteristics of an organism that which will give you guys the final result at the very end. So please don't forget the term workup. Later on, we will go back to that one. All right, what is the common precaution that is often followed when we're doing catalase test? Hydrogen peroxide should not be used directly in blood agar plates or any culture media that contains blood agar plates. We always do it as an inoculum form in an inoculum form because of the fact that blood contains catalase as well. All right. So you can't use hydrogen peroxide because the blood contains catalase. All right, and catalase is the one that produces the effervescence. That's the reason why when before in for wound care, hydrogen peroxide is being used, and a lot of the children would oftentimes prefer hydrogen peroxide over alcohol because of the bubble formation. So yeah, we don't use hydrogen peroxide in any culture media on any culture media because of this phenomenon. Because of course, blood contains catalase. All right, so please be mindful of this one because it always comes up in the board exam. Always comes up. I think even in PAMET quiz shows, I'm quite certain It was mentioned to me by my professor way back in 2000, in early 2010s, when when there was a case presentation describing 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 the medical technologist intern's performance of a specific test. The question was sent like was phrased like this: Maria was doing catalase test. Because of the absence of slides and test tubes in the laboratory, she directly added hydrogen peroxide on the plates. Which of the following would most likely render her test results erroneous? That was, I think, the question before and how it was phrased. And basically, the choices are random things that random things out of the blue, and then at the very end you'll find the answer, and the answer is she added it directly to the she added hydrogen peroxide directly to the plates. So that's how they usually phrase these questions, and it might be a shock to you guys, but it's actually, if you look at it on a different perspective, they're just paraphrasing the questions, and they're trying to confuse the students. And that's one of the ways you might be caught off guard when you take the exam. So that's the reason. I suppose this is also the reason why I'm here. I'm mentioning these things so you guys will not be caught off guard. All right. So now we're going to move on with the difference between micrococci and Staphylococcus species. So Staph species of Staph aureus in general, to be much more specific. So Staph aureus is actually aerobic. It actually grows in both aerobic and anaerobic conditions. Hence the term facultative anaerobe. All right. Micrococcus, on the other hand, is an obligate anaerobe. All right. So it's an obligate anaerobe. All right. Micrococcus is an obligate anaerobe. Grows only in anaerobic. Oops, sorry. Typographic error. Let me correct that in my, yeah. So hopefully I wouldn't be bashed because of this. All right. So wait, hang on a sec. Anaerobe. All right. So what do you mean by facultative? They can survive on both. They can survive on both things. They don't really need oxygen. They don't really need anaerobic conditions to solely survive. But when we say obligate in Tagalog, obligado na mag-follow siya ng particular conditions. So, obligate anaerobe, obligado siya na walang hangin. Alright? So, yun yung parang, shortcut ko doon to differentiate these two terminologies. Because I think I didn't mention what's the difference between facultative and obligate in my previous, did I mention that one, guys? I'm not sure. I'm not sure if it was mentioned in my previous lecture, but hopefully you guys still remember what's the language that we're using, so you guys won't be confused with it. Did I mention it last week? Let's try to ask Joanna. Joanna, did I mention what the difference between obligate and facultative organism? No, no. What about fastidious organism? Did I define it to you guys clearly? Joanna. I'm Hoy, parang diniscuss ko yon. Yeah, I mentioned it. No, I mentioned it before. I think. Uh, Hasek group. Did I mention Hasek group? Hay, nako, hindi ka nakikinig siguro last week. Let's try to ask Adrian, our lovely moderator for today. Adrian, did I mention?
Yes, sir. Na-mention it last week.
All right.
Ano po?
Okay, good. Good. What about fastidious organisms? Did I mention did I mention them clearly last week? Okay, wonderful. All right. So technically somebody's keeping tabs. Unfortunately, at that moment, I suppose May Joanna wasn't paying attention to me that much. All right, Joanna, Joanna. All right, gumising ka. All right, now let's go to drug susceptibility test for identification. So again, there's another thing that you need to remember. So aside from novobiocin, for gram-positive organisms, you need to also remember lysostafine and bacitracin or bacitracin. I keep on hearing other professors say bacitracin and others say bacitracin. Let's just focus on the spelling. Or in this particular lecture, I'm going to, and even in subsequent lectures, I will say bacitracin all the time. And please don't be a hater. All right. So lysostafine, from the name itself, it will lyse staphylococcus, and therefore it will be having a sensitive result. All right. On the flip side, micrococcus will have resistance to lysostafine. The opposite can be said for bacitracin. Why? Staphylococcus is resistant to bacitracin, while micrococcus is sensitive. All right. Now, another way to differentiate them is oxidase test. Sir naman, ang dami ko naman kailangang memorize. Si staph at saka si micrococcus lang naman pala yung pag-uusapan. Yes, please guys, please don't forget. Microbiology is actually one of my favorites because this is one of the subjects where you actually feel like you're a scientist. Okay? So yes, oxidase test did, sir. Yes, you have to do oxidase test as well. And in this particular case, you have to do a modified one. All right? So the oxidase reagent originally has tetramethyl paraphenyl diamine dihydrochloride. I know it's a mouthful, but um, when I was studying, I had to remember these chemical names or the formula. So please don't forget the reagent used for oxidase test is tetramethyl paraphenyl diamine dihydrochloride because later on my Kahoot game or later on, you will have questions regarding chemical names. So I'm excited. I'm excited how many people will be able to answer these questions because that will be our Kahoot game later. All right. Now, modified oxidase, on the other hand, aside from the tetramethyl paraphenyl diamine hydrochloride, we will have dimethyl sulfonamide as an added reagent. All right. And what are the results for the following organisms? Well, because Staphylococcus is a facultative aerobe, it will have a negative test. Whilst on the other hand, Micrococcus is a anaerobe and therefore it will produce a positive result. Similarly, when we go to Pseudomonaceae family, you will see these tests come up again. All right. Okay, so please don't forget the chemical names for the following test. You will also have, I think, another one later para amino dimethyl benzaldehyde. I love these names. I don't know why because I feel like a scientist when I keep saying them. But yeah, whatever. I think I should have taken up pharmacy because I like saying chemical names. But I think let's go to the class if they like chemical names as well. Um, what about let's talk about it. Um, who's in the class? Do you like Mayra? Mayra Gagazo? Are you on the line? Interview. Interview lang tayo saglit. All right. Do you like memorizing chemical names? No? Oh my gosh. Wonderful. All right. What about Charis? Pag need lang po. I know, right? I think pati sa clinical chemistry, you're not only going to memorize you're not just going to memorize the chemical names, but you're also going to memorize what happens to the chemicals after undergoing some sort of enzymatic reaction, blah, de blah, 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 all right? What about Arisgado Lindell? I think Lindell is a new addition to the class. I haven't heard from her for quite some time now. I'm not sure if is she actually a student here. I've never seen her in the class in the participants. But yeah. What about AJ Aquino? AJ Aquino said, "Sorry sir, I was late. Naputol pa kasi yung signal." All right. So AJ, do you like memorizing chemical names? All right. So he's quiet. Well, let's try to ask one of the another person again. Rainiel, do you like memorizing chemical names? Because later on, I already gave you a clue. You will be memorizing. If you you will be answering questions in Kahoot that is that is solely about chemical names and formulas. All right. So let's go back to our lecture. All right. So what is the metabolic function of the metabolic mechanisms followed by the following organisms? Staphylococcus, from the name itself, from the initial reactions with mannitol salt, agar, it is a fermentative organism. All right, hence it follows fermentation. While micrococcus, because of its obligate aerobe status, it's oxidation. All right, okay. Hence, it is the one that will test positive for oxidase test. All right? I know. Yeah. All right. So now let's move on to the test that detects that's the most important pathologic determinant for Staphylococcus aureus. It's actually coagulase. All right. And the test used to determine it is coagulase test. We have two types of coagulase tests. Please don't forget the slide coagulase method and the tube coagulase method. What's the difference? Slide coagulase detects cell-bound coagulase, while tube coagulase detects free flowing coagulase. Really, sir? Yes. There's a difference. Okay? Now what are the reagents that what reagent can we use for coagulase? Well, we can use rabbit plasma or serum. Sorry, we can use rabbit plasma. I don't know why I wrote serum before when I was a student. See, I remember the errors when I was still a student. But you're supposed to use plasma, not serum. I still remember it vividly. Pa rin. All right, so let's go back to that question and remove serum because when I was young, I didn't know any better, I suppose. Okay. Really, sir? Truly, sir, I have to kill a rabbit, sir? No, this is manufactured. Don't worry about it. But if you don't have rabbit plasma in the laboratory lying around, you can actually use citrated plasma. You can't use EDTA mainly because it it has calcium on it. No, it has EDTA and it can chelate and can basically interfere with the coagulation process. And citrate is actually used because this is actually a coagulation related study. And anything that's used for coagulation studies, regardless of whether or not you use it for microbiology, it will you will most definitely come back to blue tap or light blue tap. Okay? So anticoagulants that we can use citrate as we can use EDTA, but again we can use citrate. Okay, which one is preferred in the laboratory because of its availability? EDTA, but again we can use citrate although it might cause false positives on some strains. All right. So in in hindsight, you have to use EDTA because it's practical and it's available. But there are some instances or some strains that may cause false positive results. Citrate on the citrate is avoided most of the time in clinical laboratories because of false positivity. All right. Now, maximum allowable time to perform tube coagulase is 4 hours, and the test should be checked every 30 minutes. Okay? There is one case presentation I remembered when I was still a student. They're going to ask you they are going to present the case such as this. Jose Jose did tube coagulase test before in a hurry. He forgot the coagulase reagent. He forgot that he was doing coagulase test. What do you think would be the error in this particular? The timing of the specimen, the incubation, the length, the amount of inoculum, blah blah blah. So the best answer to that question is the amount of time. All right. This is how they're going to phrase the questions in the exams. So I'm giving you guys all the necessary information you need when you have to answer questions in the board exam. Now, why is it important to allow to why is there a need for the maximum allowable time when performing coagulase test? Because there are some strains of organisms or of sorry of staph. There are some strains of staph that's going to cause a false negative result, and you need to be mindful of these organisms because they produce fibrinolysins. Okay, so that's the reason why we want to complete the 4 hours. All right. Now, since we talked about the maximum allowable time for the whole test, in order to confirm negative coagulase results, you have to leave it for more than for 20 hours and store the result and at what temperature it has to be stored at 4 degrees Celsius. All right? So you have to confirm it. So when you get the positive result, yay, good, whatever, fine. But when you get a negative results, you have to confirm your negative results by storing it at 4 degrees Celsius 24 hours. Where is the 4 degrees Celsius? Refrigerator, of course. All right? So please don't forget this. This is important. Confirmation. I think it's under quality control, quality assurance of tests. Okay? So what may be the cause of false positive coagulase? Now we're moving on to coagulase. Now you have to be mindful of false positive coagulase results. Um, this is mainly due to citrate and citrate quanticoagulant plus citrate utilizing organisms because they can release because of the release of calcium. The calcium will then initiate the basically the function of calcium in the whole coagulation cascade is that it's a cofactor to a lot of things, correct? And it will activate both intrinsic and extrinsic. Some cases, this will result into a false positive result. And in some cases, this will result into a positive result. Sorry. Okay, so the cause of a false positive coagulase test is a citrate utilizing organism. And the reason why is because you used a citrate anticoagulant. And if those two things are combined, they will have the release of calcium, and this results to clump formation or clot formation. Basically, you can actually determine if it's a false positive from looking at the tube, but you have to be experienced with this. All right, you have to be experienced in looking at it. Complete clumping of I think 90% of the serum will be clumped. Sorry, of the plasma will be clumped in true positive results. But for citrate anticoagulant or citrate utilizing false positive results, the results are a little bit fishy. They will have pieces of clumps together because the organisms are not that much. All right, and the release of calcium isn't that much, so they will have like small pebbles or small clumps of coagulation, of coagulated material at the bottom. So that's for me that I think is one of the things that they taught us when we were training in microbiology, how to spot, how to also spot false positive and false negative results. All right. So now let's move on to coagulase positive species. There's a lot actually. And how do we differentiate them? Well, let's start first with coagulase positive staph species. So we have Staph aureus subspecies Staph aureus or sub Staph aureus Staph aureus, Staph aureus aureus, okay? And then we have Staph intermedius, Staph hyacus, and Schleifleri subspecies coagulans. All right? So there's four Staph species that are of clinical importance. Okay, coagulase positive staph. Let's not forget Staph aureus subspecies aureus. All right, Staph intermedius, Staph hyacus, and Staph schleri subspecies coagulans. All right, Schleri pala. Sorry. Oh my gosh. Na ano na din ako, nangongo na din ako. Sorry guys. Now, the test to differentiate all four of these is tube coagulase, Voges Proskauer, and PYR test. PYR, again, is another test that we're going to talk about, pyrrolidonyl test. Okay, so basically, tube coagulase will only test positive for Staph aureus. Voges Proskauer will be the other one, and PYR will be for one specific. We'll see all of them together. All right, all of them will test positive with. Sorry, all of them rather will test positive for tube coagulase, and some of them will have variable results. And this is what it looks like. Boom. Alam ko, masakit sa ulo. Yes, guys. Kailangan memorizing, sir? Yes. Sorry. So Voges Proskauer is going to differentiate Staph intermedius and Staph hyacus. It will not differ. It will be used for. It will test. Staph hyacus will actually test negative in most cases for Voges Proskauer and PYR. For your information. All right. So these are the results. Please be mindful of these results because you need to remember this, especially if it's going to be for biochemicals. All right. Now, let's move on to DNA's test. Really, may another test again? Yes, you have to use DNAs. All right. So another one, and this could be a little bit controversial because the only organisms that will test that will test DNAs will be three organisms: Staph aureus, Serratia marcescens or Serratia species, and Muraxella or Branhamella species. All right, Muraxella branhamella, other names just as. All right. So the contents of the media is basically it's going to have DNA in the media or the nucleic acids in general, and then the positive result is green. All right. If the organism has DNAs, it will clear out the zone around it. All right, because it has methyl green, it will remove methyl green. So basically, this is the answer to that particular slide. Please don't forget other organisms that will test for DNAs, Serratia and Moraxella slash Branhamella. All right, but I don't think anymore. I don't think Branhamella appears anymore because they reclassified it as Moraxella. So I removed it yesterday. Okay. Now DNA's media has a positive result, clearing. All right, it will clear a zone around it. It's colored green, and if it becomes clear, that's the time, that organism is DNA is positive. Okay? Now, what about protein A? Protein A is another thing that you need to remember. Since we're talking about pathogenic determinants, DNase, coagulase, this time the next one is protein A. We have to talk about protein A as well because going to be responsible for it's going to be responsible for the inhibition of phagocytosis. Why does it do that? What's the action of this immunoglobulin? It naturally absorbs the FC portion, rendering the FC portion of the immunoglobulins, rendering rendering opsonization useless. All right. So usually bacteria are opsonized, but because of protein A, it absorbs the FC portion and it neutralizes the action of the opsonization, and therefore you're going to have your the if the organism has been opsonized and then the opsonins have been neutralized, and in this case it's the FC portion of the immunoglobulin, then there's no the phagocytes around the human body will not recognize the organism as something to be targeted, or even other cells around the world, the human body around the world tuloy, around the human body. All right. So now let's move on to the enzymes that are released by Staphylococcus. Please don't forget the following: Strap, staphylokinase or staphylokinase, DNase, hyaluronidase, lipase, and coagulase. Okay, and these are the actions of the following. DNase basically destroys DNA, lipase destroys lipids, hyaluronidase is the spreading factor. This is what causes the organism to spread around, all right, around the body. Okay, and breaks hyalin basically, or connective tissue. And then staphylokinase is a fibrinolysin which breaks apart organisms. Okay. All right. So now let's move on to toxins that are released by Staph aureus. We have exfoliative. Okay. So um, uh, one way for you guys to remember the diseases associated to Staphylococcus is understanding the action of the toxins. Exfoliatin causes skin degradation. Hence, you will have the scalded skin syndrome, or SSS. No, it's not the social system. No, no, it's not that. It's different from the SSS that we have in the Philippines. All right. Another one is leukocidins. Basically, it prevents the actions of the leukocytes, and hemolysins causes what we expect, beta hemolysis, and staphyloenterotoxin will cause toxic shock syndrome. All right. So these are the things that you need to remember. All right. Another name for toxic scalded skin syndrome is Ritter's disease or Ritter's disease. Now again, based on base it on the spelling, not on the pronunciation. All right. Yesterday lang yun, yung Schlichter test. All right. But in this particular discussion, it doesn't matter because you guys have a lot of things to memorize. Okay, so remember the toxins, remember the diseases associated with them, because most of the time they will enter, especially toxic shock syndrome. One way of finding out whether or not an organism is associated with toxic shock syndrome is they will present it like this. Patient went to the went to a seafood restaurant, or rather went to a restaurant, and after eating for quite some time, the patient suddenly started vomiting for 2 to 4 hours from eating at the restaurant. um, stool coliforms were noted as negative with rapid testing. Salmonella serology was negative, and culture and sensitivity for stool analysis are to follow. Which of the following would most likely be the cause of the patient's diarrhea, food poisoning? That's how they would present it. And usually the one that has the fastest effect in the human body is actually the one that is called the type of food poisoning that has the fastest effect in the human body is actually Staphylococcus aureus. Okay? So 4 to 6 hours is a quick way to suffer from the effects of food poisoning. And that's because of the enterotoxin produced by staph. Okay? So we have another thing that we need to talk about. Leucocidins. All right? It is another name for this one is Pontu-Valentine factor or the Pontu-Valentine factor. All right? And I guess that's the end of Staphylococcus. Now we're going to move on with Streptococcus. Okay, so now Streptococcus is classified in different ways. There's a lot of classifications. We have Landsfeld classifications on the serological classifications, but in this case, we want to talk about Landsfeld classification. This is based on the antigenic group specific cell wall polysaccharides. So if we're talking about the antigenic group specific cell wall polysaccharides, we're going to talk about it, or we're classifying Streptococcus based on the Landsfeld classification, which is, I think, still the conventional way of classifying Streptococcus species. All right, and the pathogenic strep belongs to Group A under the Landsfeld classification. All right. And I need you guys to remember this one because it comes up in the board exam. Which of the following is the pathogenic, is the, which of the following Landsfeld classifications would most likely be pathogenic to man? And they would say group A, group B, group C, group D. Sometimes they would mess with your brains and they would go group A, group B, group C, and then group G, something like that, just to confuse the students. But always remember group A under the Landsfeld classification is the pathogenic one. All right, and can you give me the pathogenic Staphylococcus that we're talking about here? Let's try with Noreen because I haven't heard Noreen's voice for a very long time and I kind of miss her. All right, pyrogenous, pyrogen. All right, sorry. All right, you weren't speaking clearly. But thank you, Noreen, for for the correct answer. I thought she's playing Genshin Impact as well, girl. Kaya naloka ako kasi sabi niya pyrogenist daw. All right. Anyway, mamaya na. Mamaya. Let's go to the most common medically important staff under Group B, Landsfeld classification. Aside from from that one, we have a Streptococcus viridans, which is basically associated with neonatal infections. All right. Then another one under the Landsfeld classification is Group D, Streptococcus, which is basically Streptococcus faecalis. They are also known as enterococci. All right. They are also known as enterococcus. Okay. Now, classification based on hemolytic patterns. We're going to have alpha, beta, and gamma streptococcus. Sir, meron na naman. May ganito na naman tayo. Yes. Do we need to remember this? Yes. Do we need to remember what it's called? Yes. What is it called then? If we're classifying based on hemolytic patterns, it's known as the Brown classification. All right. So we need to indicate the species of strep that are um, that are associated with these things. So with the hemolytic patterns. So alpha hemolytic strep are going to involve the group A. The alpha hemolytic strep will include Streptococcus pneumoniae, Streptococcus viridans, or and some group D Streptococcus. And then for beta hemolytic strep or your pathogenic strep group, you will have ABC and some groups of D. Now, an important thing that you need to remember in Brown classification, usually they're going to ask which of the following species are involved in the beta hemolytic streptococcus. Which of the following species of strep is beta hemolytic? You need to remember Streptococcus pyogenes and Agalactiae. Actually, Streptococcus agalactiae sometimes, most of the time rather, is alpha hemolytic. But because of the length of the incubation, they're they're oftentimes classified as beta hemolytic because of their hemolysis. The hemolysis are actually fast acting, and therefore, unlike the other species of strep, they would give the impression of alpha hemolysis of beta hemolysis, but in actuality they are actually beta hemolytic strep. Okay, so this is actually a misnomer, but because of the 24-hour routine standard, the scientists actually classify them as beta hemolytic strep. Okay, but in actuality, Streptococcus agalactiae need to be differentiated from Streptococcus pneumoniae because they're both alpha hemolytic organisms. And this is the reason why Brown classification or the Brown classification of Streptococcus is rendered obsolete. Okay? Now, based on gram stain morphology, describe the appearance of Streptococcus pneumoniae. It is bullet shape, it is flame shape, it is lancet shape. So this is the appearance of this one. Okay, and I do not take credit for the photo. Unfortunately, I wasn't able to name who it was because it was such a long time, and copyright wasn't a what they call this copyright wasn't um, implemented back then. But this was from a slide that my professor gave to us from way back 2010 or 9. All right, so this is the appearance of this one. So I do not take credit for this one. This is from my professor's slideshow. All right, so lancet shape, bullet shape, and flame shape. That's the appearance of your Streptococcus pneumoniae. All right. And actually, guys, you will see that if you take the ACP exam, they will give you a picture of this organism plus the biochemical and the clinical case presentation of the organism, and then you're going to choose which of the following organisms are the most likely cause of the patient's disease. Because as we mentioned before, examiners cannot distinguish the fine line between medical technologists and clinical microbiology specialists or infectious disease specialists, which are basically doctors. All right. Now, describe the colonial morphology of Streptococcus species. You will see in the picture that it has pinpoint colonies. Oh, why is there no picture? Oh my gosh. Picture for this one. I think it got deleted when I changed the color of when I changed the design of the slideshow. I'm terribly sorry if there's no color, there's no picture, but I remember there was a picture for this one. I'll try to show it to you guys later on. All right? Please remind me later. All right? Okay. So now let's move on. Okay, so now let's move on to the two medically important beta hemolytic strep. Okay, medically important beta hemolytic strep. We have Streptococcus pyogenes and Streptococcus agalactiae. Again, Streptococcus agalactiae is actually alpha hemolytic, but because of the 24-hour routine testing, sorry, because of the 24-hour incubation period, they are classified as beta hemolytic strep under the Brown classification. So please don't forget, Streptococcus pyogenes and Streptococcus agalactiae need to be differentiated with each other. PYRase or the PYR test, pyrrolidonyl test, will test positive for Streptococcus pyogenes as well as bacitracin. All right, CAMP and hypurate hydrolysis will test positive for your Streptococcus agalactiae. Okay, now let's talk about PYRA's test for a while. Okay, so the reagents that we're going to use for Streptococcus pyogenes as PYRS, sorry, PYRase will be pyrrolidonyl acrylamidase. Okay, pyrrolidonyl acrylamidase, and the color developer is paradimethyl amino cinnamon actually is my mnemonic for this one. All right, pyrrolidonyl, aryl amidase, and the color developer or the color indicator is paradimethyl aminosinimaldehyde. All right, cinnamon. Cinnamaldehyde is actually the aldehyde that you can that you can smell when you have cinnamon, and that's my cue card there. That's my cue there. Ah, it's talking about PYR, the color developer for PYRAs. PYRAs. I want to go to every time I hear PYRAs. Um, After this lecture, every time you hear about PYRAs, I want you to remember Cinnabon. Why? Because the food in Cinnabon has cinnamon, and the color developer for that one is paradimethylamino cinnamaldehyde. And the positive result is colored red. Ang layo, color green kaya sir ang color ng ano? Color green kaya sir ang box ng Cinnabon, light green. Huwag na kayong makulit, Christmas naman. Red ang packaging ng Cinnabon. Alright? Wag niyo nang anuhin, wag niyo nang questionin yung mnemonic ko. Basta tandaan ninyo, cinnamaldehyde yung ano niya, may para dimethyl amino cinnamaldehyde yung color indicator niya na ang box ng Cinnabon because the lecture is being conducted this time, the positive result is red. Alright? Alam ko walang kakwenta-kwenta yung ano ko, yung mnemonics ko, pero sana matandaan ninyo. Alright? Now, let's talk about the catalytic reaction of PYRAs. So basically, you will have levopyridone, okay? And because you have PYRAs, which is pyrrolidonyl, it will break it up to two forms. It will break it up to two forms. Levopyrrolidonyl beta naphthylamide. Sorry, I didn't move the slide. I was thinking ahead of time. Sorry. So levopyrrolidone will be broken up by PYRAs into two forms, into two things: levopyrrolidonyl and beta naphthylamide. Now this one, which is your beta naphthylamide, will be the one to react with paradimethyl amino cinnamaldehyde, which is your color developer. All right. Now let's move on to another test, which is CAMP. Okay. CAMP basically stands for the type of the people who discovered the test or developed the test, or the identification of. Sorry, was I muted? Okay na, okay na. But you guys got got the idea uh, of pyrrolidonyl, right? You got my explanation of pyrrolidonyl or the PYR test, right? That was clear to you guys. Okay, so we can move on to camp. Camp test is a name for is basically if we transcribe this, it would be basically Christie Atkins, Munch, and Peterson. So it's a name of random scientists who've developed the test, and it happens to come up as camp. All right. So the organism used in this test is your Staph aureus, and yes, you use a control organism. Okay, and the positive result that you will notice in camp is it will produce an arrowhead hemolysis. Really, sir? What does it look like? Well, as I mentioned before, Streptococcus agalactiae will be beta hemolytic, correct? And aside from that, um, Streptococcus pyogenes is also beta hemolytic. And Streptococcus agalactiae will be the one to test positive for this one, and the appearance will be like this. All right. So the first streak that you see on top, do you guys see the mouse? I'll go and use a highlighter. Highlighter is here. So you guys see this portion over here. This one over here is your control organism. This one over here is your test organism. Okay, so this is a presumptive. Look at the appearance of the test organism. It produces somewhat gamma hemolytic colonies, but with the appearance of, with the combination of their growths, Streptococcus agalactiae and Streptococcus and Staphylococcus aureus, they would produce this phenomenon, and that is the arrowhead hemolysis that I'm talking about. Now, let's talk about high purate hydrolysis. All right. So high purate hydrolysis requires two types of reagents, sodium hypurate and ninhydrin solution. All right. And the positive result for high purate hydrolysis is purple. And the organism that will test positive for high purate hydrolysis is Streptococcus agalactiae. All right. Oops. The slide require that we need the reagents, sorry, again, the reagents that we need for hypurate hydrolysis is ninhydrin solution and sodium hypurate. The positive result will be for the positive result will be positive. It's streptococcus.
Sir,
Purgang
choppy
purga.
ka.
Yeah, I think so. But I don't think it's the internet. I think it's my ano. It's okay now. Okay, thank you. Sorry for that one. All right. So let's answer the last bullet. Which beta hemolytic strep tests positive for this one? It's Streptococcus pyogenes. Streptococcus agalactiae rather. All right. So the answer to the last one, Streptococcus agalactiae. Okay. So next, let's move on to hypurate. What are the hypurate products? Well, basically the end products of hypurate hydrolysis. I'm not gonna go on with the um, chemical chemical composition and chemical reactions associated with the hypurate hydrolysis test. But I want you guys to remember the end products of hypurate hydrolysis. It's benzoic acid and glycine, right? So benzoic acid and glycine. And of course, lastly, I think one of the tests that we need to remember is reverse camp. So reverse camp is basically the opposite of this one. Okay? So this one, in this case, sorry, let me remove this one. Remove this one. Omit Streptococcus agalactiae and remember Clostridium perfringens and Clostridium remember Clostridium perfringens in this case. Okay, so we want to identify another organism, Clostridium perfringens, with reverse camp. Okay, and one of the tests to do that is and one with reverse camp, and one of the organisms we want to use it in is Clostridium perfringens. Now this time you're using Streptococcus agalactiae as your control organism. Okay, the results are the same, but the test organism is different. Hence the term reverse camp. Now, this time, our test organism is Clostridium perfringens, or an unknown organism presumptive to be Clostridium perfringens. When I say presumptive organism, it hasn't been identified yet, but based on the initial biochemical and morphological characteristics, we can assume that this might be Clostridium perfringens in our workup. Okay? So when we do that, we want to confirm it's actually Clostridium perfringens, hence we do reverse camp. All right? So I hope the language that I'm using isn't jargon. All right? So please don't forget some of the language that I'm using. Um, Work up, presumptive, because these things will appear again and again, even in the board exam. All right, so please don't be mindful of those one. Work up means the process at which or the algorithm that we follow. For example, earlier we have we have Micrococci versus staph species. How do we differentiate them? All right. And then another one is another one in our workup is all right. We identified the organism as staph species. Now, next thing that we do in our workup, according to the algorithm, we need to test first with either with MSA, with fermentation methods. All right, it became positive to that one. So that's basically a workup. All right. So you understand what. Okay, clear, clear, Adrian. Oh, okay. Hmm. Anyway, let's try to repeat it again. What do I mean by work up? When I say work up, I mean identifying the test organism or the unknown. All right. So when I say presumptive, that is the one that when I'm using this language to us to that I've already assumed the type of organism that I have. All right. So it's not to confuse you guys. All right. Now let's move on with the answer to this question. Um, Reverse camp. The organism that we want to identify is Clostridium perfringens. The modification in this modification, what organism is used or what test organism do we use? Streptococcus agalactiae. And what is the positive result again? The positive result is uh, arrowhead colonies or arrowhead hemolysis. All right, now let's move on to sulfomethoxazole trimethoprim sensitivity test. It's a long, it's a long, long name. But we want to use this test to differentiate them from the other Langsfeld group of organisms, which is basically Streptococcus, group C Streptococcus. All right. So yeah, that's just another nice to know question. It's usually not asked in the board exam, but it's a nice to know question. Sulfomethoxazole trimethoprim sensitivity test. And the one that would test resistant to this one is group C Streptococcus. So I'm not gonna dwell deep into this because usually the Strep C organisms are your Streptococcus mites, the one that are usually found in the teeth or in the mouth or the oral cavity. All right. So now let's move on for the tests for scarlet fever. All right. So guys, hindi siya bastos ha. Baka mamaya, before I move the slide, hindi siya bastos. Don't think this was a malicious attempt of me to don't think this was a malicious test because the names of the test are as follows: Schurz, Charlton, and this one. All right? So guys ha, hindi siya bastos. I already gave you a disclaimer before I started with it. All right? So based on susceptibility test is it's the first one. I'm not going to say that one because I might be I might be put into capital jail if I say it. Okay? Medyo ano na tayo ngayon kasi medyo lit na ngayon ang schools. All right? So this is the test for susceptibility and Schuh-Charlton is based on neutralization. All right? So what are these tests basically? These tests are immunoassays. Okay? Now, the two tests for the two tests for scarlet fever. So we use erythrogenic toxin on the patient. Basically, we introduce the erythrogenic toxin for the patient, and then the positive result would be rash formation. And then in short Charlton, again, it's blanching phenomenon. Basically, blanching phenomenon is a diagnostic marker for scarlet fever. Is this going to be asked in the board exam? Because this is a clinical test rather than a laboratory test. Um, we're not sure, so we added it here so that you guys have at least an idea of what are these tests. Okay, and yeah, so inunahan ko na kayo ha, hindi siya bastos. All right? Erythrogenic toxin ang ginagamit natin dito. All right? Get your minds out of the gutter, children. Charing, children talaga. All right, so am I still choppy, Adrian? Or okay, okay. All right, okay. So, kasi sabi mo choppy, because maybe because I was discussing uh all right. All right, terribly sorry. All right, so now let's move on to infections associated with strep pyogenes. Strep pyogenes na naman, sir. Lagi na lang strep pyogenes ang pinag-uusapan. Yes, because a lot of the questions in the board exams usually come from staph and strep. Please, guys, if you want to review on some, if you want to focus on something in microbiology, if you don't have enough time, if you focus on staph and strep, I assure you, you might get some points, even if you didn't know the other ones. But again, there's Enterobacteriaceae, which is another one that we're going to talk about later on. All right. So, what are the infections associated with Streptococcus pyogenes, which is Group B Streptococcus, by the way, Landsfeld, not the Landsfeld, the other one, the Group B. Basically, the infections are associated as follows: sore throat, skin infection, scarlet fever, and post-streptococcal infections. So when I say post-streptococcal infection, this is what you call sequela. All right, sequela is basically the term used for um, the transfer of a disease from one side of the body to another. All right, so sequela is an important thing that you need to remember because the sequela of Streptococcus pyogenes will either go to the kidneys or will cause a disease known as rheumatic fever. Okay. Now the pathogenesis of this one is basically associated with the immunoglobulins being produced against Streptococcus pyogenes. All right. So in glomerulonephritis, the antibodies produced by the antibodies produced against Streptococcus has a similar antigenic site in our kidney cells, in specifically the glomeruli, and therefore it causes glomerulonephritis. And in rheumatic fever, okay, it actually targets the part of the brain that causes fever, hence the term rheumatic fever. Rheumatic fever, scarlet fever, blah, blah, blah. These are the things that are oftentimes related to Streptococcus pyogenes. Now, I want you guys to remember that Streptococcus pyogenes has six pathogenic determinants. Yes, there are six. So please don't forget about these things. We have hemolysins. I'm gonna close my eyes because I know I can remember it when I close my eyes. Hemolysins, streptokinase, streptodomase, streptodomase, polymerases such as DNA and RNAs, erythrogenic toxin, which is the one that causes scarlet fever, M protein, and I think I'm missing one. Wait, hang on a sec. Hang on a sec. Let's try to move it. I think I'm missing one. Diphosphatidine dinucleotidase, which basically kills WBCs. All right, so I still remember five of them. So hopefully you guys will remember six. All right, so these are the actions. Hemolysins are basically the same. Will hemolyze the cells. All right, and then streptokinase digests fibrin. Streptodomase clots blood or platelets. If you want to sound posh, and then M protein has its own function because we're going to talk about M protein next. Now, M protein is another one. So, another protein na naman. Remember last time may pinag-usapan tayo A protein. There's another one. Yes. There's a lot of these things that you need to remember. They didn't tell this to us when we were applying in medical technology school, but or clinical laboratory technology school. But yeah, you have to remember a lot of proteins that are classified as one letter only. All right, so we have M protein, A protein, what else? So please don't confuse them with your other subjects because there's other other ones that have these kinds of terminologies as well. So M protein is found in your Streptococcus pyogenes rather. It's found in Streptococcus pyogenes, and it causes the following. There's four: clumping of platelets and WBC, prevents chemotaxis, prevents phagocytosis, and it's a fibrinogen factor. All right. So there are two. So there are another. There's another thing that causes clumping of platelets. Please don't forget them. Streptodomase. All right. So M protein is different because it's a combination of many of them, plus the addition of prevention of phagocytosis. All right. Now let's move on to the hemolysins. I did put hemolysins on this slide because there's a specific topic for them altogether because we have what you call the SLO and SLS. SLO is basically streptolysin O, which is the one that we use in the one that we detect in serology. This is the um, oxygen labile streptolysin. And another one is streptolysin S, which is O2 stable or oxygen stable. Now, how do I differentiate if the organism or the strain of Streptococcus pyogenes that I'm testing is actually producing streptolysin O and streptolysin S? How? How do we do it? In the laboratory. It's not included in the slide and it's going to be a question. It's going to be a question for the class for 100 because I know this is a difficult question to answer. Your clues are in the your clues are in the slideshow right now. Oxygen labile and oxygen stable for 100 pesos. GCash. Oh, 100 pesos mataas ang bayaran this time. My goodness. Paskong Pasko. Yes, because malapit na ang birthday ni Sir. Oh, 100 ngayon. For 100 pesos, who can answer to me the most practical way of testing streptolysin O and streptolysin test? I'm not asking the specific name of the test. I'm asking you guys the procedure in which we can screen for streptolysin O and streptolysin S. Can anybody wants to try? 100 peso load on the line before we go to break. I'm not asking about the specific test, Charisse. I'm asking for the actual procedure. ASO, yes, you're correct. But I want to know if you guys still remember your bacteriology lab. How do we test for oxygen labile and how do we test for oxygen stable hemolysis? Before ASO was created, this was the way bacteriologists were determine whether or not the organism is SLO producing or SLS producing. So anybody can try. I'll give you guys 5 minutes because if you don't answer this one, we're going on break. 5 minutes on the clock. Pwede na ako mag-break, Sir Marco, no? All right. Pero before I go, I'll give the class 5 minutes to answer this question. All right. So anybody can answer. Unmute yourself, okay, and explain to me how would we be able to detect SLO and SLS in the laboratory traditionally. Sir, ano po ba ang hemolysis? Ano po hemolysis sa SBA po? Of course, in SBA, sheep blood agar. Sir, vertical stab po in culture. Vertical stab? All right. Explain to me the principle. I want you to explain to me your principle. Because vertical stab, there's a lot of tests for there's a lot of vertical stab tests in culture, but you need to explain to me why it is so. Adrian. Mhm. Uh-huh. 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 Okay, so in that okay, so you're almost there. Oh my gosh, you're almost there. 3 minutes on the clock, huh, Adrian? Adrian, you're almost there. Now, how would I find how would I determine whether or not the organism is producing SLO or SLS in your procedure? Yes, you're almost there. Oh my gosh, you're almost there. Now, how would I know? Release. 3 minutes on the clock. Saan? All right. So I'm asking now, how would we detect SLO and SLS? You're almost there. Oh my gosh, you answered it correctly actually, but the details are incomplete. All right, who wants to try? There's still 2 minutes on the clock. Anybody? Raise your hand and then unmute yourself. 100 pesos Gcash is on the line. Don't worry, Noreen, I still haven't forgotten. Don't worry. Baka sabihin ng mga students kasi, sir, scam ka, no? All right. We still have 1 minute on the clock. Anybody else can try? All right. We still have 1 minute on the clock. Anybody else can try? Noreen, can you try? Maybe it will increase your grade in your increase your prize money to 150 if you can try. What about Rainiel? Pang-load sa Genshin Impact. May timer tayo, oh. May timer. All right, Adrian. Ah, what? Heat. Oh, sino doon si streptolizin S, sino doon si streptolizin O? Oh, now differentiate it for me. You're almost there. You actually have the ID. You actually have the idea. Just so happens, I think you're focusing on what you're reading from Google or whatever you searched. Oh, you're focusing on your try to use your analytical mind. Critic. Saan? Time's up. Time's up. Sorry. All right. Before we go, I'm going to give you guys a little bit of an. He's actually correct, but the details of it is not understood. It's not understood by the people, I suppose. Did you understand it? The explanation of Rainiel, Noreen, Mayra. Did you understand this explanation, or was it incomplete for you? Because if you understood it, I would give him 100. I don't want to be unfair. I don't want to be labeled as unfair. Baka sabihin ayaw lang magbigay ni sir. Did you understand it, Noreen, Mayra, and Rainiel, based on his explanation? Or no? Ay, quiet silang lahat. Walang nagkoko. Wala sumasabotahe sa kaklase nila. I love it. Napaka ano nila, no? Pero ikaw na lang, sir. Ikaw na lang. Kasi ikaw ang ano ko, ang unofficial judge ko. Based on his explanation, did you understand how vertical stab can help you identify whether the organism is producing SLO and SL? Uh, Uh, but but were you able to but were you able to deduce what he was saying and detecting whether it's SLO or SLS? Kulang ang details, di ba? Um, all right. So, sorry Adrian, si Sir Marco na ang nagsabotage sa iyo kasi ayaw magsagot na mga kasama mo. Ayaw sumagot. Siya yung pinakamalapit. Oo. Oo. So I think I would give him a preliminary. I will think about it. Let's think about it later, Sir Marco, no? Let's try to I'll think of. Oh, you're going to be you're going to be my judge officially because apparently these students are so loyal to each other that they're not going to risk looking like a jackass in front of other students. All right? So, sorry for the term. Uh um so what Adrian was saying, I'm gonna help out Adrian here. And this is the surface of the agar. All right. Sorry. Okay. So this is the surface of your agar plate. I'm gonna put a black one on there. Ayaw talaga nilang ayaw talaga nilang isabotage yung performance ng kasama nila. I appreciate it actually. But in this case, it's considered as Hunger Games. Charing. Actually ako na yung hungered talaga. Pero because Adrian was able to cause mental stimulation mental stimulation with his example, I will try my best to explain it to you guys. Okay? So this one, and then we're gonna go with fill tool. Oops, red. Okay, so imagine that's the surface of your culture media, and then this is your this is a streak of your stab, as mentioned by Adrian. All right, so that's your inoculating needle. All right, so that's vertical stabbing. If you're not familiar with it, that's a procedure that they traditionally used. Imagine the colonies are in the bottom part of the agar of the culture media. Okay, so wait, still having difficulties trying to control this thing. All right, so that's your inoculating needle, and then you've made a stab. Oh, oops. You made a stab, right? The agar, because of its formation, because of its jelly consistency, it will block the entrance of air. So air won't be present in this portion of the culture media where you produce your stab, where you made your star when you made your where you made your stab. Okay, so here is your inoculum. Okay, that's your inoculum. So this one, after you pull out um, the needle, it will close up. Okay, it will close. Sorry, let's change the color to red. It will close because the consistency of the agar is solid and it's jelly. Because of its jelly consistency, it will somewhat close impartially. All right. So what would happen is that as the organism grows, if the organism has SLO, which is what are the clues again? The clues are as follows. SLO is oxygen labile. Okay? Oxygen labile. If the organism is producing SLO, let's go back to paint. This one, this one will cause beta hemolysis. It will cause beta hemolysis on the culture media. And you will see it because you need to stab it on the edge of the agar. All right. So this one will produce beta hemolysis, and it will be characterized as this one. If it's if it's oxygen stable, if it's oxygen stable, it will produce hemolysis along the whole stab area. How do I know this? How will I know this? You need to do the stab method on the edge, near the edge of the media, for you to assure yourself that the stabbing method is the hemolysis pattern is observable. All right. So is it clear, guys? If it's SLS, it will produce something like this because again, based on our definition, it's oxygen stable. It will not be destroyed in the presence of oxygen. If it's SLO, it will be destroyed in the presence of oxygen. Is it clear, Adrian? That is the one that I'm asking you earlier. That's the one that I'm asking you earlier. How would I know whether the organism is producing SLO and whether the organism is producing SLS? Is it clear, Mayra? Wala na. Quiet na si Mayra. Gutom na rin. All right. So it took me 15 minutes to explain it to you guys because of the if I had a chalkboard in front of me, it would have been faster. But because of the pandemic, we have to at least try our best on explaining it to you guys. All right. So is it clear? Can we eat now, guys? All right. All right. So may I ask for an hour this time? Is it fine with Mr. Marco? An hour. All right. Thank you so much. Because I have to cook my own meals today. No, there's no delivery today. So thank you so much for graciously allowing me to take a break time. Thank you. All right. So this is your timer for an hour.
Ano, no? Of our lecture.